When I see a tree without leaves in the winter, its intricate branching architecture reminds me of a similar, much smaller tree that looks just like that. But the tree I'm thinking of is actually inside of your liver. The biliary tree, as depicted on the right, is an arboreal network of tubes that stretches to the edges of your liver lobe and drains bile, a metabolic waste product, from your liver into the intestine. However, if the biliary tree is absent or not fully formed, then the bile can't drain, causing extensive tissue damage and eventually organ failure. My focus is a disease affecting the very formation of this biliary tree, called Alagille syndrome. Children born with this condition have biliary trees that are absent or severely truncated, and 3/4 of these patients will require a liver transplant by the age of 20. But what if, instead of relying on a liver transplant, we could regrow the biliary tree from the inside? To do this, we turn to a phenomenon observed in our laboratory. We use a mouse model of Alagille syndrome that mimics the human disease. So our mice are similarly born without biliary trees, as depicted on the left. But here is where I must reveal a small secret, because the fully fledged biliary tree you see depicted on the right is actually from this exact same kind of mouse that was born without any biliary tree at all. Unlike humans, this mouse can spontaneously generate a biliary tree equivalent to a normal mouse. Surprisingly, we found that just as a seed completely transforms in order to become a tree, the primary liver cell type, called a hepatocyte, completely transforms its cell identity, fate, and function in order to generate the biliary tree. Now, in my role as a gardener scientist, I'm unearthing the mechanism behind this by isolating individual cells of the biliary tree at discrete stages of its development. Using computational tools, I can visualize how the genes expressed by these cells fluctuate over time. So far, I've identified the genes responsible not only for the change in cell identity, but also the structural change in the size, shape, and organization of the biliary tree branches. Right now, I'm testing to see if I can accelerate this process by delivering the genes I identified to the seed of the biliary tree itself, the hepatocyte. If successful, this approach represents a tangible strategy for inducing, directing, and maintaining the growth of this essential structure in humans. Thank you.